talk even more about mitochondria actually. And I would like to ask our next speaker, Michael Hammond, who's a postdoc at Biological Center at České Budějovice, who will be talking about mitochondrial genome and evolution.
Yes, thank you. So let me just get started. So I will talk about the complexity of the Glenogracillus mitochondrion as revealed through proteomic studies. So most modern eukaryotes possess a mitochondria or a mitochondria-related organelle. It's well known for performing oxidative phosphorylation to generate ATP, but the mitochondria has many other roles other than that. It is important for iron sulfur cluster synthesis and can initiate programmed cell death. So 3D imaging of Glenogracillus has shown that its mitochondria is a single reticulated and connected organelle. So I will refer to it as the singular mitochondrion from now on. And as pointed out in the last presentation, it's a dynamic organelle that responds to aerobic and anaerobic environments, clustering around either the chloroplast in aerobic conditions and around the cell membrane in anaerobic conditions, producing wax esters, which are of biotechnological interest. It has a somewhat reduced mitochondrial genome, even compared to other mitochondrial genomes, only encoding 7 proteins. And so this means that, like all other mitochondria in modern eukaryotes, the vast majority of proteins has to be imported from the cytosol into the mitochondria. And this means that genome and transcriptome studies are of limited use when trying to determine the total complexity of the mitochondria. So we employed sonication to disrupt Glenogracillus cells and then separated them via sucrose fractionation, which generated clear fractions corresponding to various organelles. We then digested the mitochondrial fraction and analyzed it via mass spectrometry. And this gave us a list of 2,704 proteins. Now, to distinguish between which proteins are genuine mitochondrial and those that are contaminants, we employed 3 strategies. First, we compared the mitochondrial enrichment when compared to the chloroplast and whole cell lysate fractions, and included those that were enriched, obviously, in the mitochondrion. We compared work to 4 other well-established mitoproteomes of T. brucei, yeast, mouse, and Arabidopsis thaliana, and included orthologous sequences. And finally, we included proteins that had strong functional annotation indicating likely mitochondrial function. And so this ended up with a total of 1,786 proteins that we deem to be genuine components of the mitochondrion. Now, what stands out about this number is how large it is. So initial estimates on the mitochondrion of Glenogracillus had estimates of about 1,100 proteins, of which this exceeds quite a bit. Established literature suggests that animals in general have approximately 1,500 proteins in their mitochondria, whereas some plants may have as many as 2,500 proteins. But importantly, when you look at the established yields that have been experimentally verified from other model organisms, Glenogracillus also stands out. So this begs the natural question of what is influencing the size of the Glenogracillus mitochondrion. So when we look at functional annotation, we see the largest enriched categories are core metabolism, ribosomal synthesis, and protein transport and folding. But ultimately, we observed that 57% of our recovered proteins were annotated as hypothetical, and this speaks to the need for increased functional studies in the proteins of euglenids. We also isolate the influence of the chloroplast on the complexity of the mitochondrion. So in plants, it has been well studied that photosynthesis-related pathways and proteins are often present in the mitochondrion, and this is also observed in Glenogracillus when you see the Galler shunt, which complements the TCA cycle, as well as sulfate assimilation, which was likely initially inherited from an ancestral plastid. Additionally, we've isolated approximately 200 proteins found in both the mitochondrion and the plastid, which we expect to be dual localized. A third factor is the extensive set of duplicated proteins. So various pathways in the mitochondrion have multiple copies for each step. And initial analysis of the flagella proteome indicates that it is also of substantial size when compared to its protist peers. So it may be that the organelle proteomes of Glenogracillus are more complex in general. So other interesting mitochondrial features, as mentioned by Mark Field, we observed no mitochondrial post-transcriptional editing of the mitochondrial transcripts. And this is interesting because the sister clades of diplonamids and kinetoplastids have their own very complex and baroque means of changing up their transcripts. So it would appear that in addition to having the smallest mitochondrial genome, that the euglenid mitochondrion is the most conventional of euglenozoa, size notwithstanding. We've also additionally identified 192 mitochondrial transcription termination factors. So these are proteins that were initially discovered in the mitochondria of other organisms and later discovered in other organelles. They traditionally have roles in transcription regulation as well as ribosome biogenesis. We have recovered a number from the mitochondrion as well as a number from the plastid, which would suggest that, like other organisms, these MTEFs are partitioned between other different organelles. Now, importantly, studies in plants and other photosynthetic protists have indicated that MTEFs confer resistance to abiotic stresses such as light, heat, and salinity. So these MTEFs may be playing a role in the general resilience and adaptability that we find in Glenogracillus. So the protein import machinery is essential for any mitochondria, as it's responsible for translocating the proteins from the cytoplasm across the two membranes of the mitochondrion or into the intermembrane space or into the membranes. So looking at the components of Glenogracillus, the most well-investigated mitochondrion import system has been in T. brucei of Euglenozoa. And Glenogracillus seems to indicate a larger, slightly expanded set, though it also contains several subunits that were previously assumed to be trypanosome specific. And in fact, when you compare this just looking at the protein pore-forming units on the membranes, while trypanosomes have reduced their pore-forming subunits to essentially the bare minimum to get by, euglenids have expanded and retained theirs, with duplications seen in TIM17 and TIM23 of the inner mitochondrial membrane, depending on the species. So we additionally have compared Glenogracillus mitoproteome to the transcriptome of Eutreptiella gymnastica, and we've identified approximately 1,100 sequences, which we expect to be present in the mitochondria. Now, incidentally, this is what was initially predicted in Glenogracillus, and this has also enabled us to illuminate the various features that we expect to be present and unique to euglenids, and those that are shared in Euglenozoa. And these include certain ribosomal subunits and the alternative oxidase pathway. So for future directions, I personally would like to see a number of experimentally verified euglenid mitoproteomes to complement this study. There are a number of in silico predicted proteins in the mitochondria of Gracillus that still require verification. We need the functional characterization of a number of hypothetical proteins, and we would additionally be interested in observing how the mitochondrion responds to changing environments at a proteomic level. So thank you.